Welcome to SciAnts Streams. If you are interested in all things science in a fun, wholesome environment, you are in the right place. SciAnts is a unique husband and wife team of Belinda and Lita, who hold their PhDs in molecular and systems biology from Dartmouth. We do a variety of interactive science streams, including microscopy of ancient amber with insects, 3D printing, art, science news and deep dives, the occasional game, and more. If you've ever wanted to have a fun space to nerd out, now is your chance. We stream daily science content on Twitch. Understanding not just what we know, but how and why we know it is the essence of scientific knowledge.
Do you know what determines the process of triggering that cast? No, Chris. No. That is what we need to do the extra experiments for, which maybe let me just go ahead and we'll go over here. We'll pull up the drawing pad because I don't know how better to explain it. Always interested in this type of information. I still owe on my master's degree, so no doctor for now. I can mooch up here always. Bots, ask the questions. That's why I'm here. Right now we're talking about how to identify the different genetics of leafcutter ants as they're developing. Your question, right, is you go from an egg, you go to a larva, then you go to an adult ant, and those have different castes, and they look different, and they act different, so it's this transition. It's not the egg to the larva. It's right here that we want to figure out. So what you need to do is there's a whole layer of complexity on this chat for the experiment. So it is not just as simple. I'm doing well, Amber. We're wondering how that caste requirement is decided. So Chris, I'll try to answer that too. Red is nurse. Green is cutter. And blue is soldier. This is very simple, Chris. There are many more castes in leafcutters, but we're going to keep it super simple. You want to figure out how do they decide which of the numbers to make. Like, what do they make, this one, or this one? So the developmental time is actually ordered in this way. So this is the fastest in terms of development, and this is the slowest. So you're talking 3 months for these, 3.5 to 4, and 4 to 5 for the soldiers, all in terms of months. That means that the stage that takes the longest is the larval stage, not the pupal stage. So it goes egg, larva, pupa, and then adult. The pupa and the egg, that's 1 month and 1 month. This is the variable component here, is that larval stage. And that, if the longer you stay as a larva, the more energy you're going to be required to fed, right? So the nurse has 3 larval stages, the cutter has 4 larval stages, and the soldier has 6. So a colony will not create or decide to have soldiers near the beginning of the colony's health because they simply don't have the resources for making soldiers. And so there's what we think, and it hasn't been experimentally proven, Chris, to figure out how this works on the genetic level, but it's a density question. It seems very much like once the colony hits a certain number, you'll start to produce the other castes. So roughly, that's why soldiers usually aren't seen in the colony until about 2 years of colony age. And that's if the colony is growing at a healthy rate, then it'll start producing soldiers. What you can do, Chris, and they've done this, it's just a lot of the labs that do these experiments don't have the funding to do RNA sequencing, which is the necessary experiment to do to ask the question that you're wanting to know is, okay, how does it work genetically? How do they know, right? Is what you can do is, let's say you take we have here a 5-year-old colony. So that has all the castes. And we take a subset of it let's say just queen plus nurses, right? So at that stage, she's been producing nurses and cutters and soldiers, but you take a small batch of it, like a really small block, and you separate it out. There are at this stage gonna be some soldiers that are gonna hatch, because there's also embryos in here, right? But if you wait, the next batch that are laid are going to be back to nurses and cutters, and you're going to have another gap before you see soldiers. So possibly a biomass trigger the preset proportions of production of each caste. Yeah, exactly. And there's some flavors of, like, not just food, but also, like you said, it's biomass of, like, the other castes as well. Maybe in the entire colony, once it starts to, like, get big enough, maybe only 15 to 20% of the colony is going to be soldiers. Because not only do they need food when they're developing, they also eat some as adults, but they don't eat the food directly. They are fed the food by nurses. So not only do they take the extra time of development and being fed, but also as adults they be fed. So they do a lot of good for the colony. They defend it, but it's a lot of extra work. So one thing you can do, I've inadvertently done this, is that soldiers are, because they're so different, you can do sequencing on them, and so that makes them like a good comparison to the nurses, right? Like, because the nurses are really small, the soldiers are really big, there must be genetic differences or expression. Kidara, thank you for the lurk, ma'am. Appreciate you. There must be differences in signaling, and so that's a good thing to compare, right? If you remove a lot, then the colony will start to produce some more to kind of make up for that loss. But you're looking at a 4 to 5 month pulse before the next batch will come out, and there might be a little bit more in that next batch. And they're constantly hatching, right? So it's not just like, pulse. Like they're constantly being produced. It's just that this pulse there might be a higher production and hatching, because they're trying to keep up their numbers, because there are, the soldiers will have about a 2-year lifespan, and so they're constantly producing to make sure that the ones that are dying are being, like, brought back, in terms of the total numbers. I hope that makes sense. So much to learn, so excited to yeah, no, it would be, Chris. You'd have to do, I think, a lot of sequencing to, like, look at what the density effect are and see how it changes with time. And you might also have to do it on queens, which makes it very difficult, because you cannot mate a queen in the lab. You have to catch them in the wild. You need a lot of replicates, but it would be amazing to study. Asking for, like, what Bots was wondering about, like, okay, what are the genetics that determine this? So, like, even if there's some food at the beginning, what happens later on? You'd basically need to harvest samples at each of these larval molds. So there's L1, 2, 3, 4, 5, and 6. And so the soldier, goes at all of these. The nurse goes through L1 through 3. And the cutter goes till about 4 or 5. And so these molts are changes over sizes. So they shed, like, they molt, right? They shed the outside layer of cuticle, and because they become too big for it, and it's just a time for them to grow. Nerding out over this hive minds tweak in my imagination. Nice, Chris. But yeah, always. It's very sci-fi, but I think it's super, super cool from the genetic perspective. And so what you'd want to do is at each one of these time points, you'd want to do sequencing experiments for each of these castes, and then compare what the differences are. So you want to look at the RNA. So you can look at, presumably, well, it would be difficult, Chris and Bots, of what you would want to harvest. If you want to harvest, like, if you had all the money in the world, if you had all the money in the world, what you would want to do. So if this is our larva, there's a larval brain and, like, a ventral nerve cord. There is a gut and, like, a stomach too. There are cells called fat body. The fat body's where a lot of metabolism happens, and they also have energy storage there. And there's a lot of protein being created and a lot of transcription and RNA. So there's a bunch of activity on that front. There's also occasion, like if it's a queen, there could be a wing disc, 2 wing discs. Those are going to be the future wings. There's other things in there as well. So then, do you, as a scientist, what do you do the sequencing on? What do you measure the changes in genetic rate with? Do you do the sequencing on the entire organism? So do you grind up the entire larva and do the sequencing? Or do you dissect and look at different tissues and individually sequence each tissue? Now, if you had unlimited money, I would suggest doing each tissue, because it might not be like the main, maybe the most important tissue that is causing the difference is metabolism based, and it's in the gut. Presumably there's a lot of changes happening in the brain too, right? But what if it's the gut that's telling the brain of where to become and how to become? And so that's like an extra layer of complexity. And how do these different tissues signal to each other, right? There's all kinds of signaling that happens here. And so if you grind up the entire larva, you'll not get that resolution in that image. Probably the starting point that you would do is do whole body, whole larval sequencing on all the stages, and then do the comparison, right? Compare the different castes and see how they're different, what the differences are. So if you compare the RNA to each of these castes and compare how it looks different. So let's draw our little graph here. And we're just picking a random gene. Let's say the random gene is really, really high in the nurse. It's low in the soldier, and it's in the middle in the cutter. Maybe that's an important gene that influences the caste identity. It probably isn't that easy. It probably isn't a single gene that's acting on it. But in order to identify it, you have to do those sequencing experiments. The next question is going to be, what does the DNA look like at that particular gene? Is it in a closed state? Meaning, is it wrapped up? And so you want to look at the differences in that particular gene. This is just to say, I don't know what it is. Jason. It's the gene Jason. Okay, we're going to call it the Jason gene. Okay? Jason is very high in the nurse, in the middle of the cutter, and very lowly expressed in the soldier. So we want to look in the genome and see what is the state of the genome around the gene Jason in each of these castes. That's an experiment called Hi-C, where you're looking at the 3D orientation of the DNA and what does the genome look like in 3D space. So a very simple way of doing that, of explaining this, would be, let's say if this is our nurse, our cutter, a soldier, and then we're going to draw out the DNA. Presumably, if the Jason gene, which we'll say in purple, is on, it'll be open for the nurse. And so I'm just going to indicate with this little arrow that it's going to be turned on, and you can make a bunch of the Jason protein. A lot of the mRNA, right, which will then be turned into protein. And that maybe makes nurse. The opposite would be true, you'd expect, for the soldier, where here is the Jason gene and it's wrapped up in this ball, and so you can't actually turn it on, and so you'll have no access to it. So just if you think about it like this, it's in this wrapping loop. This is where the RNA polymerase needs to come, but it can't actually get in there because of the loop. And if it can't get in because of the loop, then it can't turn on that gene, and so you'll get no Jason. No Jason for Jern. The cutter is probably something intermediate, if it's getting intermediate levels. It could be a variety of things. It could be that, it's a little bit of a loose, like 3D space. It's not fully wrapped so it can still get in and transcribe, but it's maybe a little bit blocked off. And so to get at that question of how it's 3D oriented, you have to do that ChIP-seq experiment. And that will give you the answer. Those two in conjunction give you the answer. And so you can look at this at a particular site in the genome, or you can do ChIP-seq in the entire genome.
Thanks for watching SciAnts Media, and be sure to hit those like and subscribe buttons to help our channel grow and support our science outreach and education goals. We can be found on Twitch most days of the week, and be sure to join us here for our next video. Thanks again, and stay curious, everyone.